Hello, everyone, and welcome to today's Cryo-EM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High Resolution Cryo-Electron Microscopy Program. My name is Christina Zamani. I'm a scientist and training liaison at the National Center for Cryo-EM Access and Training, NCCAT. Here with me today is my colleague at NCCAT, Ed Eng. Joining from the Stanford SLAC Cryo-EM Center S2C2 is Michael Schmidt, and from the Pacific Northwest Cryo-EM Center PNCC is Omar Devolku. Our speakers today are Shubham Dubey and Michelle Bush from the Department of Biological Sciences at Purdue, and I just realized I'm meant to be sharing my screen, so let me do that. Thank you. And I will introduce our speakers properly after my colleagues from each of the centers give a quick update on the resources that we make available to the community at no cost. And this webinar is a regular event. We host the last Thursday of the month where we highlight the methods and give tips and tricks that the groups who are using the national resources collect and interpret the data they collect using National Center resources. So you can actually save the date already for our July talk. PNCC will be hosting Kenneth Childers at Western Washington University, who will tell us about his work on coagulation factors. So you can register for that talk already on the events page at cryoemcenters.org. And on that page, you can also find recordings from all of our previous talks. For Zoom logistics today, I will be moderating questions at the end of the talk, but you can send them in anytime using the Q&A box, but please use that Q&A and not the chat. And so first, I'm going to turn over to our center representatives to give us a quick update about what's happening at all of the centers. And first, I'm going to kick off to Mike.
Thank you, Christine. I'm Mike Schmidt. I'm one of the co-directors of the S2C2 Stanford SLAC Cryo-EM Center. As with all the centers, we have the mission to provide high-resolution data collection facilities for people to collect data on their high-resolution samples. We are committed to training crystallographers and other structural biologists to be fluent in cryo-EM. So we have a complete training program which is ongoing, both on site and remotely. And we have a screening service for people that might not have screening microscope capabilities to make their data ready for high resolution data collection. So come visit us. This is our website. And we hope to see you either for training or data collection services or for screening. Thank you.
Thank you, Mike. And now I will turn over to Omar.
Thanks, Mike and Christina. I'm Omar Devolku. I'm one of the microscopists here at the Pacific Northwest Center for Cryo-EM in beautiful Portland, Oregon. We share the same mission that Mike just described. We offer our users a seamless workflow from sample preparation to 3D reconstruction. Briefly, in terms of hardware, we have 4 Titan Krioses in Arctica and shared time on a Glacios, all of which are equipped with K3s, fringe-free imaging, most of which have Gatan energy filters. We've also got a number of Falcon 4Is and a cold phage source. In addition to collecting data, we also offer many training opportunities, both remotely and in person, on topics such as sample preparation, usage of serial EM for screening, data collection, and data processing. And the last thing I want to mention is our screening access mechanism, in which users are invited to work directly with us here at PNCC to collaboratively freeze, screen grids, and collect data. So I'd encourage anyone with questions or anyone who wants to apply for time to either reach out through the Q&A or come to our website, pncc.labworks.org.
Thank you, Omar. And I will turn over to Ed.
Hi, my name is Ed here in New York. Again, like the other centers, we offer screening, training, and access. On the access front, this summer we'll round up our last FFI upgrade, then we'll offer all our Krios instruments with FFI. On the screening front, we welcome you to try our grid preparation screening program. You can use Chameleon, a blot-free vitrification device, to help you ameliorate certain issues with standard plunge freezing. Or if you'd like the standard screening process, a center staff member will help you with plunge freezing and screening. Our next call for proposals is July 1st, but given the holiday, we'll extend that. If you would like to make use of that extension, please contact us at the NCCAT user office. And you can find out more on our website at nccat.nysbc.org.
Thank you, Ed. And before I introduce our speakers, I wanted to make a quick plug. If you want to talk to us in person instead of on Zoom, we're traveling to a number of both smaller regional as well as large meetings over the next couple of months. In fact, this week, there are staff from our centers at the ACS Northwest regional meeting. That is a fresh picture from yesterday, if any of you are in the Northwest region. And also before our next webinar, we will be at the exhibit hall at both the ACA meeting taking place in Baltimore, as well as the M&M annual meeting in Minneapolis. And we're especially excited to talk to investigators who want to make use of cryo-EM, but need help navigating the resources. So if you'll be at any of these meetings, come say hi to us, send your friends to come say hi, and talk to us about how we can help you become cryo-EM practitioners. One final shameless plug, a workshop that I'm involved in organizing, the ACA meeting, which is coming up next week. I'm co-organizing with center staff from X-ray beamlines and other structural biology resources on how to make the best possible sample for experiments using an array of structural biology techniques, including small angle X-ray scattering, crystallography, and our favorite cryo-EM. And the registration deadline for that meeting is this Friday. And if you're already registered but hadn't thought about attending a workshop, you can contact the ACA main office and still register for workshops through Friday. And I think one thing that maybe not everyone knows is actually the ACA is really a home for cryo-EM. There are multiple cryo-EM sessions as well as workshops. So it's a meeting worth checking out. So I'm going to stop sharing and introduce today's speakers. So with us today are Shubham Dubey and Michelle Bush, who are both graduate students in Nicholas Neunach's lab at Purdue University. The lab studies proteins on the outer surfaces of pathogenic bacteria, where they gain structural insight in order to aid novel antibiotic discovery efforts. I asked them a bit about what they enjoy about the science they do. Michelle's favorite part of working in the lab is being able to unravel mysteries of pathogenesis by understanding the function of microscopic machineries at the molecular level. And she's also a seasoned science communicator and has hosted a microbiology educational video show where she talked about infectious diseases and the importance of microorganisms in our daily lives. Shubham's favorite part of working in the lab is that her studies of proteins essential to pathogenesis may have significant real-world implications. And she also enjoys the challenge of interpreting the mood swings of the bacteria she cultures, trying to keep them all in a good mood so they express her protein. So she works hard to keep her bacteria happy. We work hard to keep our microscope happy. And together we do some beautiful science that the two of them will tell you about today. So I will hand it over to you.
Okay, I guess I'll start by sharing my screen, which I hope I share the proper one. Just let me know if everybody can see it.
Yeah, that looks great.
Okay, perfect. So thank you to everyone for being here and for having us as well. It's a privilege to be able to transfer a little bit of our knowledge and our science to you guys. So we hope that you enjoy it. So I'll start just talking a little bit about my project, and then Shubham will continue in the more science involved part, since she is the one that has more experience with cryo-EM. But I wanted to share a little bit about my project because we were able to collect some interesting data in NCCAT. So my project focuses in studying the interactions that occur between the beta barrel assembly machinery complex and SURE, which is a chaperone protein. And this complex is important in gram-negative bacteria because it's essential for holding and introducing outer membrane proteins into the outer membrane of all gram-negative bacteria. So it has been shown to be essential for cell viability, and its importance has been proposed to be a suitable candidate for drug as a drug target. So as you can see here, this complex consists of 5 proteins and come together, with BAM-A being the major subunit, being the outer membrane protein as well, and it has a beta barrel domain in the outer membrane. And then it has 5 polypeptide associated transport domains, also called ultra domains. And then we can also find 4 lipoproteins in this complex, which are BAM-B, BAM-C, BAM-D, and BAM-E. And then you can see here the intricate web of interactions that occur between all of the components in this system. So as I mentioned before, this complex is so important because it's able to introduce all of the outer membrane proteins into the outer membrane of gram-negative bacteria. And these outer membrane proteins play vital roles in the cell. So how does this process start? So the outer membrane proteins are born in the cytoplasm, and here they have a signal sequence that directs them to the inner membrane, where they are translocated by another protein complex called the protein complex. Once the outer membrane proteins, which is denoted by this squiggly red ribbon here, get to the periplasm, they have to interact with their chaperone proteins, specific chaperone proteins, which I like to call their dance partners. And here, these are SURE or SCIP. The chaperone proteins are responsible for stabilizing these outer membrane proteins and also transporting them across the periplasm. Then the chaperone proteins interact with the BAM complex and delivers these OMPs to the BAM complex, where then the BAM complex is able to fold them properly and insert them in the outer membrane of all gram-negative bacteria. Now, the process by which BAB introduces and folds the outer membrane proteins into the outer membrane is a process that our lab has well characterized already. So the focus of my project is in all of the interactions that occur in the periplasms, which are vastly unknown. So how outer membrane proteins interacts with the respective chaperones is unknown, and how these chaperone proteins interact with the BAM complex is unknown as well. So from previous research, we were able, or Dr. Wu, which was a previous graduate student, was able to solve a structure of the BAM complex with SURE bound to it in the presence of a substrate or an outer membrane protein. So we know by this structure the interactions that occur between BAM and SURE, which is the chaperone protein. So we can see here, SURE in blue interacts with BAM-A in green. It also interacts with BAM-B in cyan. Once again, here in the bottom, interacts with BAM-A. And finally, there's interactions between SURE and also BAM-E. Now, I was interested in determining whether these interactions are also possible in the absence of a substrate. And if SURE is able to interact with the BAM complex in the absence of a substrate, what does this conformation would look? Is it different from the previous structure? And how it affects the dynamics of the BAM complex? So what I did is that I purified the BAM complex, and then I purified SURE separately. I mixed them together and run an SEC column to allow complex formation. Then this sample was put into cryograms, and it was sent to NCCAT for data collection. So here is where this fun part starts. So our trip to New York and collecting the data. So this was the first time I was ever doing cryo-EM. I come from a very heavy clinical microbiology background, so structural biology is still all new to me. So it was very helpful to be able to go in person and just learn a lot and have all of the facilities of being asking questions and immediately getting responses. So we were able to collect 9,717 micrographs, and which resulted in a little over 8 million particles. So the data set was not that great in the sense that sample preparation could be optimized. So I had to do a lot of rounds of 2D classification to start looking at the features that I was expecting. And then after this rounds of 2D classification, I was able to get 3 ab initio models. And after 3 rounds of heterogeneous refinement, I started to see some interesting features, which I saw extra density where we expected to see density for SIR-A based on our previous structure. So after rounds of homogeneous, uniform, and local refinement, we ended up with 52,588 particles, and we were able to see 2 very distinct features from our previous structure of SIR-A bound to BAM-A in the presence of a substrate. So we saw first that the barrel domain of BAM-A was in a very different conformation from our previous structure, and also by increasing the contour of this structure that I have here, I was able to see extra density on the bottom of BAM-A, where I expect SIR-A to be present. And I saw 2 different conformations of this extra density, which I'm showing only one here in this image. So how does the model look like? So this is a top-down surface view, like if you're looking into the barrel of BAM-A. BAM-A is denoted by this green structure and this blue structure. So the green structure is a structure that was solved by Dr. Wu in the presence of substrate, and the BAM-A is in an inward open conformation. Now, in my structure, we can see that it's a very different conformation. It's in an outward open conformation, and this is in the absence of the substrate, so the outer membrane protein. Now, looking at from the bottom up, the periplasmic view, you can see here this dark blue ribbon is SIR-A on Dr. Wu's structure, and the density that I see for my structure for SIR-A is denoted in cyan and also in green. So you can see there's 2 very distinct conformations on SIR-A for my structure. So to have a better view, here is a video showing the dynamics of the complex when it's bound to SIR-A. As you can see here, there is, I call it like a breathing of the barrel. It expands and contracts. So you can see here is in a closed conformation, open conformation, and also we can see a lot of movement in the potra domains, which are in the periplasmic side, and also the swinging motion of SIR-A. Now this is another view of the complex, looking at from the periplasmic side, and you can see how SIR-A moves towards the barrel domain of BAM-A and also moves away from it in this swinging motion. So in conclusion, we see that BAM-A, so we can see that SIR-A influences a conformational change in BAM-A, possibly to make the insertion of OMPs favorable. And this is our hypothesis by possibly threading the substrate into the lateral gate, which the lateral gate of BAM-A is this here. So threading substrates, oh, I think you can't see my pointer. So threading the substrate comes from the bottom, and possibly this movement of SIR-A is threading the substrate to come up here to favor the insertion of this substrate into the outer membrane. Now, this was 3 days of very intense work. And also we were learning a lot along the way, especially me, because this is the first time ever I was doing anything of this. So we worked really hard, as I mentioned, and if you ever go to the New York Structural Biology Center, you'll see Ed taking a lot of incognito pictures. You'll notice when he's around taking pictures. So we were very excited to be here. And if I would have to go, if I would do data collection again, I would definitely do it in person. I think my PI is here, so Nick, send us again. And we also played hard. So where the New York Structural Biology Center is located in Harlem is just a perfect location. So we were able to go to the park, we were able to go to museums, we were able to go to the opera as well. And it was just a very fun trip where we were very exhausted at the end of the day, but everything was worth it. So we really thank for the training that you guys provided over there. And with that, I'll leave Shubham to continue her part. Thank you very much.
Thank you, Michelle, for handing over the screen. Christina, can I start sharing my screen?
Yes, you can.
Okay. Can you see my screen?
Yes.
Okay.
Yep.
I hope everything looks good there.
Looks perfect.
Okay. Yeah. So my name is Shubham, and first of all, I would like to thank organizers for giving us the opportunity to talk about our research here. So my PhD basically revolves around this system, which is present in the bacteria Neisseria, which helps it to acquire the iron from the host, and how it does so we will see later in my talk. So why Neisseria? Why we care about Neisseria is because there are two of them which are pretty nasty: Neisseria gonorrhoeae and Neisseria meningitis. They cause the sexually transmitted disease gonorrhea and meningococcal meningitis, respectively. So if we talk about the Neisseria gonorrhoeae, it has already been listed as the urgent threat level by the CDC. And if we talk about the drugs that are available against this pathogen, there is only one which is effective, and it is also giving us multiple drug resistance. And there are multiple case studies being reported that patients are already being resistant to these bacteria infections. And talking about the prevention side for this bacteria, currently there is no vaccine. Now I'll switch to the Neisseria meningitis, which is a little bit kinder one that can be treated. But the problem is that the diagnosis of the disease can be commonly confused as common flu. And that gives us a very short time to treat the disease. So that's why we need a better prevention against the Neisseria meningitis. And the current vaccine, MenB vaccine, is not universally effective and also serves a very short-term immunity. With COVID hitting, we all know what a short-term immunity means. So I will not go into that. But yes, by looking at these charts and data, we know for sure that we need better countermeasures against both these pathogens, Neisseria gonorrhoeae and Neisseria meningitis. So when this microbe or any microbe infects in our human body, what happens that they need some sort of common nutrients, which is of human interest and as well as of the bacteria interest. And here I'm showing this as a picture where they're fighting over the a metal, which is iron. So that is the interest of my whole project. So when microbe infects, they infect and they fight for this particular iron nutrient, and they try to fetch it from human. What human has a strategy over the course of evolution that it doesn't allow the bacteria to get its iron, and they do so by keeping them in their proteins such as transferrin, lactoferrin, hemoglobin, and etc. So these proteins not only transport the iron from one location to the other, they also make it very unavailable for the infecting pathogen. And this strategy works great when other bacteria infect in our body. They don't get iron, and this strategy works out great. Infection doesn't spread. But when we get the Neisseria infection, we got a problem here because Neisseria is well smart enough to get the iron from these proteins, and we will see why. Is because this bacteria is equipped with this surface machinery on its outer membrane, which helps it to get the iron from the human host. And can you see my cursor? Just checking.
I can, yes.
Okay, so these proteins are decorated at the surface of the bacteria And they interact with the human proteins such as human transferrin, human lactoferrin, hepatohemoglobin, and hemoglobin. And these systems can be one component or two component depending on how many members are involved from the bacteria side to do the whole process. If it is two component, then there will be a beta barrel situated in the outer membrane and also a lipoprotein seated next to it. And then these proteins together, they interact with the human protein and try to perform the whole process of iron acquisition. And if it is one component, there is just a beta barrel required for the whole process. My system of interest is transferrin-binding protein. It's a two-component system having TbpA protein, which is a beta barrel, and TbpB protein, which is a lipoprotein situated at the surface of the bacteria. And together, they interact with human transferrin to acquire the iron. This particular transporter, TbpA, is also known as TonB-dependent transporter. Except TonB-dependent transporter, it will need the energy of the TON complex, which is situated in the inner membrane to perform the iron acquisition from the human host protein. So now we know about the system a little bit. So why we are focusing on the TbP system is because all the reported pathogenic strains of Neisseria express this system, which makes it really good that if we find something against this system, we can target the bacteria and it will be translated to all the pathogenic strains effectively. And then there has been studies that if you take this system out, the bacteria has shown the compromised survivability in the host, and also the pathogenesis has been prevented. So this system is pretty much important for the bacterial survival and as well as pathogenesis. It has also been shown that if you make the antigen based on these proteins and infect them in the mice model, they show a very nice immunogenic response, which makes these proteins a very nice vaccine candidate. And that's why it's important to understand them deeply. And lastly, if we find some contacts which we can target through the drugs, it will be really nice to have more drugs against this bacteria in the future. So with these important features being shown by a TbP system, it has been there for a while. So it's very hard for us to not find anything on the PubMed. And in this case, pretty significant reports already available. The first one is a crystal structure solved by my advisor, Nick himself. So he solved the crystal structure of TbpA in complex with transferrin. And what happens when he crystallized this protein, he used the transferrin, which was non-iron bound form, just to make the whole system as homogeneous. And the second report was in the iron bound conformation, but the protein used was TbpB, which is the other protein in the system. And together, these crystal structures provide a lot of molecular details of the individual protein. So there are three things involved in this whole process: a beta barrel, TbpA protein, which looks like this, a TbpB protein, which is a lipoprotein situated at the surface of bacteria, and there is a human protein involved, which is called human transferrin, and it has iron in its N lobe and C lobe. And there will be a lot of N lobe and C lobe happening later on in my talk. So I want to give a molecular introduction of the proteins in the very beginning. So if we know all the molecular details of the proteins individually, what is yet to be done in order to target these systems? We know few molecular details, but we know very little mechanistically about the system. So the crystal structure I showed you 2 slides ago was solved with the transferrin, which had no iron in it. So we wanted to see how the structure looks like when the iron is present in the complex. If we have iron bound structure and we already have non-iron bound structure, then we can understand the mechanism of iron extraction and import. That was the question number 1. And the secondly, now I showed you that TbpA interacts with the non-iron bound transferrin, and it has to interact with the iron bound transferrin in the first place to start the process of iron extraction. So if it is interacting with both forms of transferrin pretty easily, what is leading to the dissociation of the transferrin once the iron extraction is done? that other question was unanswered. And lastly, we know nothing about at the molecular level what happens when all three components are together in the process where TbpB, transferrin, and TbpA all are interacting with each other to perform the iron acquisition. So with these initial questions, I started my work in the lab, and what we did was, like, first thing as taking TbpA and transferrin in iron bound form this time, and being said that it's a TonB-dependent transporter, so that and because of that, to get the iron from the transferrin, it will need TON complex from the inner membrane of the bacteria. And we are not providing the TON complex in the system, so we should be able to capture the iron bound form of the transferrin in the complex. And with this plan, we started sample prepping. We had some issues which we worked out with the bacteria, like Christina mentioned, that they have mood swings. So finally, we are in a stable relationship where bacteria tries to understand me and I try to understand them, and it worked out. We can purify TbpA, and this is a complex that I ended up after 365 days of this plan over here in the right. So after making the complex, we collected cryo-EM data here at Purdue cryo-EM facility. And this is just a pipeline to show what we follow in general in field to solve the data from the cryo-EM. I won't go into the details, but I'll be happy to take questions later if we have any. But the end story is that we were able to get the 3.7 angstrom resolution map of our structure, which looks like this. In the green, we have TbpA, the barrel part, and in the yellow, we have transferrin here in the top. So once we solved the structure, we fitted model, everything fitted well in our structure. We put both the components in there, and then to our interest, we quickly zoomed into the C lobe of transferrin to find the iron, and there was no iron. So as I said, it's a TonB-dependent transporter. We thought that it will need TON complex, and we are not giving it, so we should be able to capture the iron. But we didn't see the iron in the C lobe, which was pretty surprising. And this was pretty new to us. We didn't think about this way when we started the pipeline of the whole cryo-EM thing. So we thought, can we prove this by some additional method so that we have more with more data saying that the iron extraction is done just by TbpA, not required by the not TON complex was required there. So we took help of a technique called electron paramagnetic resonance. So to perform this particular technique, you need, first of all, electron, and that electron needs to be paramagnetic. That being said, that it has to be unpaired electron, and these teeny tiny unpaired electron will serve as an individual magnet, and they will align themselves when you provide a microwave frequency which matches the lower energy state and higher energy state, and electrons will start flipping over these lower energy states and higher energy state. So with this information, luckily our system itself has a paramagnetic center. The iron serves as a paramagnetic center in the HDF and in the transferrin, and that makes our system a very good candidate for the EPR studies. So we went ahead and we collected the data on the transferrin itself, and as you can see here, that at G factor 4.3, so G factor is just is a value. It's a fingerprint of a electron in certain environment. So at G factor 4.3, we see this peak right here in the transferrin, which is absent in the apotransferrin and also absent in our buffer. So if adding TbpA does something to this particular, peak, then we will know that TbpA is disturbing the iron coordination center in the transferrin. So here I'm showing you the data where we had the transferrin itself in the buffer first, and then we provided TbpA instead of buffer in the second sample, And we see that the signal has got down. That being said, that some of the iron is not there to give the EPR signal, which was present in the transferrin alone. This supports our cryo-EM observation that, yes, TbpA is doing something to the iron coordination site in the transferrin, and it's able to extract the iron from there. So if we combine our cryo-EM data and previous crystal structure and the EPR studies, what we see is that if you give TbpA, does not matter holotransferrin, which is the iron-bound form, and the non-iron-bound transferrin, you will always end up with a non-iron-bound transferrin form and C lobe in a partially open state. The N lobe state of the iron will depend on what form of transferrin you started with. If it is the holotransferrin, there will be iron in the N lobe, and if it is apotransferrin, there will be no iron in the N lobe. So in our structure, we see iron in the N lobe, but we don't see iron in the C lobe because we started with this form here. So by this time, we had good enough data, preliminary data, to start with a proposal. And thank you to NCCAT that they gave us the opportunity to collect data on. So from here onward, whatever the data I'll be showing you was collected remotely. So I would like to take a pause here and thank you to all of you who has been helping me over 1.5 years to collect the data remotely. So thank you for that. And finally, this was the time when COVID was there. So we couldn't go in person. And recently, we were able to go and mark our feed there. If you ever go to the NCCAT, you will get a pin. So you can mark your location and it changes the color, how many people have been there from that particular location. So we want to change the color of our pin. And we want to visit multiple times. So thank you for all the opportunity. And now getting back to the science part again. So I showed you the TbpA in complex with transferrin. We tried to get the iron-bound form. We didn't succeed, but we did learn something new from that structure. The second effort that we did in our data, we thought that let's incorporate the other component which is there in the system, which is TbpB. And if we read the literature of TbpB, what it does is that TbpB N lobe goes and binds to the C lobe of transferrin and make it in a close conformation so that iron doesn't escape. And this helps the system to acquire the iron more efficiently. And this was the story of this crystal structure reported here that I showed you earlier as well. So what we did that we just followed this crystal structure. We made TbpB and HDF complex first, and then we added TbpA to it. And then what we expected that we will have a triple complex with TbpB, transferrin, and TbpA into it, where we will be able to see the iron because TbpB will help keeping transferrin in the close conformation, so we can see iron in the complex. So we went ahead. I'm just skipping the data collection processing part. It was pretty much similar what I showed earlier. Jumping to the data. So we solved this beautiful, gorgeous triple complex structure where we can see all three components in there. You can see that TbpB makes a long interaction here with TbpA. There are some interactions between TbpB and transferrin. You can see TbpB goes all the way in the transferrin to do something. We will discuss that later. And then after solving this map, we fitted all the models into our structure. And the first thing we did is zooming into the C lobe. And to our surprise, there is no iron in there. So we thought that adding TbpB will help us and will give us the iron-bound structure. But it didn't go the way we thought about it. So and why it did not help us, the answer was there in the structure itself. So if we see the TbpB and HDF complex, this is what we started with, N lobe and C lobe sitting close to each other. And then we have other structure of TbpA and HDF where the C lobe of HDF interacts with the TbpA. And if we combine these two information and translate this into a triple complex, you will expect that this thing will come and sit on TbpA and the complex will look something like this. But our triple complex did not look the way we expected it to be. Instead, the triple complex is forming a C-lobe interaction, and this TbpB part here is totally flipping and interacting with the transferrin, and transferrin is in partially open state, not in the closed state, which helps us understand that why we don't see iron, because end lobe is not there to protect the iron escape from the transferrin. So structure does tell us a lot of story. We just need to dig deeper in the process. So just to show you, I showed you with models, rough models. Now I will show you what's happening in the molecular level. So if you see the TbpB end lobe, it looks like this, which has a barrel domain and a handle domain. And if you see TbpB C lobe, that also has a barrel domain and a handle domain. And if you align them on each other, they align pretty well. The barrel domain is almost very same, and they are only different in their handle domain and some of the loops. So the handle domain is what gives them identity if it is TbpB end lobe or C lobe grossly speaking. So I was, when we solved the triple complex, we were pretty much confident that end lobe needs to sit here, transferrin needs to sit here, TbpA needs to sit in the down. So we kept fitting end lobe for a while in our structure, and this is how the fit looked like for almost a week or two. And to us, it appeared that this feature does not belong to alpha helix, definitely. It looks like some barrel. And then we flipped the TbpB, and after flipping, it fits perfectly fine. Like barrel is there, some of the loops are even fitting fine, and the helix is also fitting fine in the very far region of the triple complex. So this is how the complex looks like at the molecular level from the top. So this gave us confidence that yes, C-lobe interaction is something true and happening in the structure. We just need to speculate why is this happening. So what is the reason behind this molecular interaction that we never saw before? Other than this observation, we had one more molecular interaction, which was pretty novel to the triple complex, is this anchor peptide, which is coming from TbpB. And this anchor peptide is running from the end lobe, which is the distal part of the triple complex, coming all the way to TbpA and forming an interaction there. So we looked into the residues that are involved in the interaction in that area. And as you can see, there is a very high positively charged patch present there. And there are a lot of negatively charged residues from the TbpB, and there are other residues around too. So what we did, we were trying to speculate, what is this interaction doing there? Is it required for the triple complex formation? So if we see to understand that, we did a pull down study. And in pull down, what we were doing is that we were giving the pre-made TbpA and transferrin complex on a nickel bead, and we were providing the TbpB. And in the imidazole elution, if we see if the complex forms, TbpB here has no histag. So if complex forms, we should see 3 proteins in our pull down assay. And when we did this with the wild type, we can clearly see 3 proteins on the gel. But when we deleted these 23 residues initially, we just chopped the whole region first. We don't see that middle band in our pull down anymore, which gives us the confidence here that yes, these 23 residues are actually required for the triple complex formation. Anchor peptide is doing something really significant in the complex formation. Then we planned a bunch of other pull downs where we deleted initial 16, 18, 19, 20, 22 residues. And then also we were not able to see the triple complex. So these were just some additional studies that we did after getting the triple complex. But the original question stays there, which is where is my iron? I tried not giving TON complex, I tried giving TbpB, where is the iron? So we revisited our sample preparation, and what we concluded is that we gave TbpB and HTF as a complex, which was the N-lobe to C-lobe interaction and had iron in it. And in the end, we ended up with something like this, where the TbpB actually flipped around and there was no iron, and transferrin C-lobe was partially open. So this is a lot of change happening from this particular state to getting to this particular state. So there might be a lot of dramatic changes happening on this incubation time scale. We just need to dissect this time scale into small chunks and freeze our sample to see if we can capture those fleeting states in our complex. So we did, we froze grids at 0 minute, 5 minute, 10 minute, 30 minutes, and expected that on this incubation time scale, there will be some time point where we will be able to capture the iron bound form in the triple complex. And if it is iron bound triple complex, we should also see N lobe of TbpB closer to the C lobe of transferrin as a supportive evidence. So I would again like to thank NCCAT for supporting all the madness, because I've frozen tons of grids and sent them over the years. So thank you for all that data collection help. So yeah, in the end, we in one data set, in the 10-minute incubation one, we were able to solve a decent map. And when we fitted model in our map, in the very beginning, the map looked a little different to us. There was no this kind of occlusion in the other structure, and this helper helix actually belongs to the N lobe of TbpB. So N lobe was closer to transferrin. And when we zoomed into the cavity, into the space, we saw iron there. All the coordination sites were intact and present there. And if we zoom in, if you just put the density on, this is how the density looks like at a very low contour. So yes, we got the iron in the end. That was really exciting. And now that we have the iron bound form and non-iron bound form, I would like to revisit the molecular interaction of TbpB and transferrin. So here I'm showing when the iron is bound to transferrin, how the structure looks like is that N lobe of TbpB interacts with the C lobe of transferrin, which is a classical reported interaction between TbpB and transferrin. So this is one structure we have, and the other structure that I showed previously has a C lobe interaction and has no iron bound to it. So we have now iron bound form and non-iron bound form, and between these two states, TbpB is doing some real weird things that we still need to understand. So if we compare our iron bound structure and non-iron bound structure, there is a lot of change happening here. You can see that the whole barrel is, as Michelle said, breathing. So I think barrel breathing is something we should just put as a term in the beta barrel field. And you can see that transferrin basically closes and opens, and TbpB basically provides the space for it to open and close. So this is just a morph from the iron bound form to the non-iron bound form. So that was pretty interesting to see how the whole molecular machinery is trying to orchestrate the process. And whatever I said previously, I would just combine everything in this one animation. What we think might be happening in the process of iron acquisition with this whole system is that at the first step, transferrin comes to TbpB. It locks it into the close conformation so that iron stays there. This whole complex comes to TbpA, and then TbpA itself is enough to extract the iron. Yeah, extract the iron. And then this transferrin is not the same, but a changed one with a partially open conformation. This partially open conformation does not support the classical N-lobe C-lobe interaction of TbpB and transferrin. And now, by some unknown mechanism that we don't know, TbpB has to flip around and bind C-lobe with the transferrin. And maybe this C-lobe interaction has some role to play in order to kick the transferrin from the surface so that the next cycle can reinitiate, and then TbpB dissociates from the surface. So this is what we think so far. I think there will be more add-ons as we progress through this whole project. But this is so far what we understand about this system. And just to show the same thing in a more fancier way, I have developed a new hobby to join the blender. So I highly recommend you doing it. It kills a lot of GPU, and yeah, so iron extraction happens, TbpB flips around, and then that probably leads to the dissociation of the transferrin, and then all the components dissociate and new cycle can initiate. So we have a better picture of the iron piracy, but we still have a lot of things to do. First, in my list as a grad student is to publish the current progress. And secondly, we are also planning, since we have some pull downs that are showing the significant effects to the triple complex formation. So we want to test these mutants into the Neisseria cell. We have a nice collaboration with Dr. Cynthia Corlinson. She has the whole Neisseria growth facility. She can test that mutants for us. And I have also received some monoclonal antibodies that can bind to TbpA. So that has been developed by our collaborators as well. So they don't know where they bind. So I'll be doing some structural characterization of those. So I would just like to highlight, and NCCAT people, please accept our proposal. We still have a lot of structural work to do. Our proposal is due in 2 months for the renewal. And lastly, since we have the iron-bound structure, non-iron-bound structure, so we can do a steered molecular dynamic simulation study to see what leads to the one phase to the other, and we can calculate some energetics to see what it requires to open the transferrin C lobe by TbpA. And one day when everything fall in the plane nicely, I will write my thesis and graduate. And with that, I would like to thank a lot of people, because this work has not been a reality without their help. First of all, my amazing advisor, Nick. I have learned over the year a lot of things from him, but I still feel I have still a lot to learn from him. So, thank you, Nick, for being there always. And all the Nuinage Lab crew member, all my funding sources, and also NCCAT for a lot of my MAT data collection with the time-resolved cryo-EM. Thank you so much. And with that, I told you that I have developed a new hobby to kill my GPUs. So this is a movie that, this is a picture that I made in last few days. So I'll be happy to take any questions. Thank you so much. Oops, sorry.
Thank you, Shubham. That was absolutely gorgeous. A lot of work. So I'm going to start off. I'm going to start off with a question from me, but I think that dovetails well into the question we already see in the Q&A. And do feel free to toss questions into the Q&A now while we start our chat. So how well can you see side chains in your maps, and can you use that to guess whether you have iron bound, or is it larger structural rearrangements that tell you whether or not you have iron?
I really appreciate your question because I wanted to tell this at some point and I forgot. So we actually don't need very high resolution to say confidently that we have iron bound, because transferrin is like this and it's open and closed conformation. When it is in closed conformation, these two lobes are sitting like this. And these big changes we can even see in 6 angstrom. So we can say that transferrin is in closed conformation. Most probably the iron is there. So that conformation, that bond, the coordination site between these two lobes are not broken. So that's one guess. And in our recent structure, we have decent enough like resolution. We can see side chains in the coordination site to say that yes, we have iron bound there.
Thank you. Now, there's a question from William Lanzillotta about your EPR data.
Mhm.
He
Mhm.
pointed out that the decrease in your intensity is minor, especially if
Mhm.
you're looking at a one-to-one complex. So he was
Mhm.
asking, are you looking at one-to-one? And then also asked, did you spin integrate your signal?
Okay, so I don't know much of the details of the EPR, but yes, we did not do the spin integrate. I can certainly add this in my notebook to do in future and read about what it can help me with. And talking about the minor change, the data that I showed for EPR is like we took TbpA, we took transferrin, we mixed it like in 10 seconds, and we froze the sample. So within 10 seconds, that much loss of the signal happened. And there is other iron in the N lobe, which is still there and which will be giving the signal. You are not disturbing, you're not touching that lobe by TbpA at all. So we don't expect the signal to be very flat or very low, but we just wanted to see if there is a perturbation happening in the signal because of the presence of TbpA, and that's what we analyze in the EPR. But yes, thank you for the advice of the spin integrate thing. I will certainly add it in my note.
All right, thank you. There's a question from Jignesh Kumar, and he asks how the TbpB C lobe and the human transferrin C lobe change conformation in the apo and holobound form. And then a follow-up that maybe clarifies, does the human transferrin or TbpA induce the conformation to the bound form of iron?
Yes, I think he answered his question
Okay.
himself.
Okay.
So yes, so the iron bound when TbpA is there, the transferrin is there in the close conformation. TbpA is the one which is cracking the C lobe of transferrin, making it a good conformation so that TbpB can flip around and bind to the C lobe interaction. There is no big conformational changes we can observe for the TbpB itself, other than this flipping. We don't see any change in there other than this rotation in TbpB. So I don't know, maybe he can clarify what conformation he's asking about. But yes, we just see a flipping of TbpB when it is in iron bound form, when the transferrin is in iron bound form and in non-iron bound form.
Thank you. I actually, William, I just let you on to, if you wanted to ask a question in person, and while you decide whether or not you want to do that, I'll pop in with one more question. Have you tried SPR or other techniques to try to understand binding affinities or other biochemical characteristics of your conformational changes?
Easy escape from this question will be, I am so busy solving structures. I am sorry. I haven't been to that path of EPR and ITC. But yes, this will be something interesting to do in future with the mutants. But just to give you an overall idea that I have performed over the 40 or more than 40 mutants pull down and purifying all 40 proteins, and it did not work. So before I go down to the SPR, where a lot of technicalities also involved, I want to be just 100% sure what mutants I want to take it to the next level. So yes, something very interesting to try, but as I said, I'm too busy solving structures.
Wonderful stuff. Can you hear me?
Yes, I can.
Yeah, Nick was down here for a talk a few years back. Anyway, that's great stuff. There's a lot of siderophores and receptors, as well as hemophores and receptors out there. And I'm certainly happy to send you guys some stuff if Nick wants to collaborate. Just one technical note in your intro slide. You may just want to modify it because HMBR transfers heme, not iron. Right?
Okay.
It takes the heme out of hemoglobin and transfers that into the periplasm. The
Sure,
iron
sure,
actually, yeah, the iron isn't released until it gets into the cytoplasm where hemoxygenase pulls it out.
Yeah,
But very
thank
cool
you, thank
stuff.
you.
Yeah, and there's a lot of unexplored territory here because we now know that formation of hemophores and receptors is inducing genetic changes. It's the formation of the complex itself, not the
Mhm.
transfer, which is really, really interesting. So lots of work for you to do as a postdoc then somewhere.
Sure. Yeah. Looking forward to it. Thank you. This seems like I had a pretty much like very specialized audience. They can catch in my introduction slide that I presented probably more than 5 places and nobody caught it. Thank you so much. I'll make that change.
And I'm going to quickly pop in for those who have to head off. Thank you for joining us. We at the National Centers are here to support your science, so let us know how we can help you, and we hope to see you again. If you're willing to stay on, Shubham, for one more question, I've got one more for you.
Yeah.
So it was your 10-minute time point that gave you something different, but do you think that was the particular time point, or was that sort of a game of numbers, right? Because you're using so few of your total particles for your final reconstruction. Was that just the grid that happened to get you that structure, or do you think that time point was significant?
Again, thank you so much for asking this question. And yes, it has been bugging me like anything. Yes, I don't think that's the only population in my 10-minute sample. I think there is a variety of sample. We have actually, yes, I have already started the topaz strain pipeline to see if I can find the less populated particles in my whole data set. If I can get so, maybe in some next presentation, I'll be showing that. But yes, I do believe that there are other population present in my whole grid that we cannot see by cryo-EM, because that's all we can reconstruct. This is the only population we could have reconstructed up to 4 angstrom to solve something and see something. So this was promising that we see the iron-bound conformation, but at the same time, we do believe that there has been already iron extracted and there will be some population which will show us the previous structure. But it's just that they are so much less in population, we cannot do a 3D reconstruction out of it. So, yeah, maybe in one grid, there will be a magic and we will be able to see both of them in one data set.
Well, we will do our best to get you as much microscope time as we can.
Yes.
And
I have a quick question, if you don't mind. Just one more.
yeah, go ahead, Mike.
The actual binding affinity of iron to transferrin, in the absence of anything else that's trying to pry the iron away from it, what is the actual sort of starting point affinity? Is it micromolar, nanomolar? What sort of binding strength is it of the iron?
I will let you keep guessing because it's very, very high. None of the range you mentioned, nanomolar, picomolar, it's 10 to the power minus some 29. It's a crazy like tight binder, and all of a sudden it goes to TbpA, it becomes like a swing and it opens up without any energy.
Yeah. Do you have any hypothesis of how it so freely gives up its iron? In fact, is the iron binding site completely disrupted when it's opened up? There's no the ligands, or sorry, the ligand binding residues are just completely not near each other at all after the iron's let go.
Yeah, so this will take a little bit of background. So when the iron binding site is like this, like this, one of the C1 from the C lobe, C1 sublobe and C2 sublobe, some residues from here, some residues from here, they combine together to form the iron binding site. And once the iron coordination site is broken, this does not stay like this, but become like this. So they are very far from each other once the iron is not there. It's a totally different conformation. How TbpA is able to like crack this high affinity binder and get a stable intermediate conformation that we can characterize structurally is happening probably. Okay, I don't have data for it, so I will not talk about it. I'll just keep my opinions to myself. But I feel like the moment the TbpA interacts with the transferrin, when it sits there, there is a helix which provides the support for the other lobe to just stay there, and the barrel moves and opens this form like this. So barrel movement coordinated with the sitting of transferrin exactly near that helix is probably the catch. But we need so many additional proof to say this more profoundly.
Thanks.
Thank you for your question. Interesting.
Yeah, well, thank you for sticking with us a few extra minutes to get to all of our questions. But we'll wrap up for today. And thank you so much. It's really nice for us to see what happens with these data after they leave our microscope. So that was really really wonderful to see. Thank you very much.